right, good morning, everyone. Thanks for coming. We wanted to just wait a couple minutes after to let folks find the room. This is a massive hotel. I don't know about you all, but I'm staying in the exact opposite corner of this whole complex. Thanks for coming. This is the Cytoscape Advanced Automation tutorial. So hopefully you're in the right room. We're going to ask you some quiz questions in a minute to make sure you're in the right room, even if you think you're in the right room. And that's so that we can adapt the content depending on exactly where you guys are at and what you want to get out of this. All right, so let's dive in on that topic of what we're supposed to get out of this. By the end of this workshop, you'll know more about Cytoscape and when to use it in your research projects. Hopefully you already know a little bit about Cytoscape, but again, we'll quiz you on that in a second. A big question we always have is where to find networks, and it turns out there's lots of places to get networks, so we're going to cover that in depth. And so by the end of this, you should know how to identify the relevant types and sources of networks for your research questions. And then the automation aspect, which is the focus of the whole second half of today's workshop, is about having programmatic control of Cytoscape. And so this depends on a REST interface that Cytoscape has called CyREST, and we're going to be illustrating that via R, but this can also be done in Python. And so if you have a strong preference for Python, you can follow along that way instead. And the idea then is being able to integrate this into your existing bioinformatics pipelines. Hopefully most of you are already using R or Python in your work. All right, so at a high level, this is the whole day. We have about 4 hours total,
4 hours.
including the break that we're going to take. So we're going to do a bit more introductions and concepts, including an introduction to network biology. I'm going to take you through those sections, and then I'm going to hand it over to Scooter Morris. I'm going to do introductions for a section in just a second for the guided tour of Cytoscape, and we'll see how in depth we need to go for that. And then he's going to cover finding network data, network analysis, and visualization. In these sections, they're mostly slides, but there's a few hands-on components to keep you engaged. And then he's going to hand it back over to me for the second half. The whole second half will be hands-on automation, running Cytoscape in R, going through various vignettes and examples, and then saving time for you to be able to work on vignettes of your choice or on projects of your choice. So if you want to dive in to writing your own R or Python interfaces for Cytoscape, we can help you with that today. All right, let's get to the introductions. So I already leaked some of the news. My name is Alex, and this is Scooter Morris. We're both coming from San Francisco. We both have been part of Cytoscape since 2006. We work right across the street from each other, but we actually met at a Cytoscape conference in San Diego in 2005. During introductions, we realized that we worked across the street from each other, and we've been working on Cytoscape ever since.
Actually, you worked for a collaborator of ours.
Oh.
Even more.
Yeah, sure, right.
Yeah, we worked.
Small world,
Yeah.
Obviously, and we know many of you in the audience, too. In addition to the two of us who will be doing most of the slides, we have you surrounded in the back. We have Dexter, if you want to just raise your hand. Dexter, Kay, Jing, and then Barry is there somewhere. Barry, Brett is behind him, and David. And so all of us will be on hand to help you during the hands-on parts of the workshop. And so grab anyone you see with an extra Cytoscape ribbon on, and they'll be able to, or just raise your hand, and they'll be able to help you through your Cytoscape and Cytoscape automation problems. Okay, so that's enough about us. Here comes the quiz time. We want to know about you guys. And so for this, we're just going to ask for a show of hands. You can raise your hand more than once as we go down this list about how you identify yourself. So who considers themselves a clinician? You actually work with patients. Great. A couple of you. Bench biologist. You know how to use a pipette. A couple of you. A bioinformatician. Yeah, good. Okay. Computer scientists. Good. Chemists, mathematicians, physicists, or other? Okay. It turns out this is our typical distribution. This is sort of a sideways density plot here. And you guys hit right on the mark with bioinformaticians, right? That's the bulk of you. We are trying to shift this curve, or at least broaden it towards the top to cover more bench biologists and even clinicians. So I was glad to see a few of you there. There's always a few chemists and physicists. You're always welcome, but
As long as there's no mathematicians, I'm good.
yeah, okay.
They check me on my math. It's really scary.
This is the scary part of the presentation. We always worry that if the whole class raises their hands for a mathematician and nothing else, we'd be in trouble. We're not going to go too deep into graph theory here. Okay, great. A couple more questions for you guys, just so that we can tailor how much time to spend on different sections. So this is gauging your prior experience. So prior Cytoscape experience. Who has none at all before? Okay. We'll be doing the introduction to Cytoscape slide then, I think. No problem. You've used Cytoscape a little bit, but you still have a lot to learn. Okay, good. That covers almost the rest of you. You do a lot with networks, say using iGraph or other network packages in R and Python and NetworkX, but not Cytoscape. A couple, okay, good.
Okay.
You do all the above. You know Cytoscape well, you know R, you know Python. I saw a couple hands, okay. You guys might be able to teach this course, so, okay, so. You're shaking your head, so maybe you regret raising your head. Okay, good. Hopefully there's not too many in this category as well. Otherwise, we'll just ask you to walk around and help others when they get stuck too. And
we'll put you to work. It's fine.
Okay. So let's dive into the slides. I'm going to start with just a general introduction to network biology concepts. This is basically to give you guys network biology, systems biology. These are very broad terms, and so this is also helping to define the terminology, what we mean by network biology, what we care about when we're looking at network graphs, so that we're all using the same terminology. Okay, so why networks? Why, as bench biologists and as bioinformaticians, why should you care about networks? So here's the initial sales pitch for networks. First, on the left here, networks are everywhere, especially in biology, right? Molecular networks and pathways that we saw in textbooks from high school and undergrad on, these are all networks. That's what we're talking about when we're talking about networks. But at the higher level, there's also cell-cell communication networks. Those are becoming more and more relevant, especially with single-cell measurement technologies becoming easier and cheaper. At a larger level of systems, right, the nervous system, they often talk about pathways and networks through brain systems. And then social networks we all know about, and those are important for epidemiology and other biology topics. So those are all networks. And Cytoscape, for example, treats them interchangeably. It's not focused on only one of them. It's most often used for molecular networks, but it can work for any of these levels of networks. Okay, and then on the right here, networks are also powerful tools. They're not just descriptive illustrations, they're also useful as research tools, and I'd argue especially in biology. They reduce complexity. They're more efficient than tables. So in a lot of your work as bioinformaticians, you're often working with data frames or tables or matrices, and we'll show a lot of examples where a network visualization or a network model of that exact same data is more efficient, and you can do specific types of analyses with it, and very importantly, you can do you can visualize it in a way that's much more intuitive and easier to communicate principles than by just showing tables. Let's start with, I like to show just one example. We have a Tumblr feed we'll show later of network examples that are published, and I just pulled this out of there. In this one network view, let's see if I can point here. There we go. In this one network view, we have a lot of different data types, so it's a nice illustration of how networks are used for data integration and visualization. So at the center here, there's a ribonucleic protein. So a protein that binds to a lot of different mRNAs. The blue interaction lines point to the mRNA. These are the gene labels, and in their mRNA form, they bind to this protein. These are all apoptotic related genes in circles connected here. Fine. But also, there's a whole nother layer here. These blue squares are transcription factors, and the green lines indicate the regulatory actions of these transcription factors on these same yellow circled mRNA apoptotic genes, right? So even without seeing the labels here, this sort of view gives you an easy way to look through the different relationships, the different groupings and clusterings of apoptotic genes, transcription factors, and this centrally acting ribonucleic protein. Now, if you think about it, the data that went into making this, right, lots of different experimental types from different labs over many years, right? It's not just one experiment produces this. So it's complicated data spread over lots of places, all brought to bear onto a single figure. Again, trying to show this in table form, which is where the data came from originally, would be pretty useless. Trying to just describe this in the paper without a figure would be very difficult as well. So we take it for granted, but these sorts of network visualizations can be really powerful for integrating data and communicating data. Okay, so the two themes we're going to come back to again and again throughout this are analysis and visualization. In analysis, we've got topological properties. We'll talk more about those later. You've probably heard of node degree, for example. That's the sort of thing we're talking about. These things help us find hubs and subnetworks. In larger, more crowded networks, it's sometimes nice to be able to zoom in and focus on subnetworks that are most relevant based on your data. You can also do classification, clustering, and diffusion on network models, and like I mentioned already, data integration. So these sorts of activities then lead to different types of visualizations, which then lead back to different types of analyses. So it's often an iterative process going back and forth between analysis and visualization. For visualization, we're talking about things like data overlays, right? So here they're using fill color to show different categories. But they're also using this red color to indicate an up expression, an overexpression of upregulation of specific apoptotic genes. And so you can turn experimental data types into visualization properties. Visualization also refers to layout. This layout isn't the one and only layout for this network. You can apply different algorithms. You can apply different manual layout strategies to better communicate the point you're trying to make with your data, as well as animation. We'll show an example of that later today. Visualization is also important just as part of exploratory analysis as well. Okay, so once you get an eye for it, you'll start seeing networks everywhere. You'll see them on the covers of journals, and you'll see them in papers used almost constantly. And so I wanted to pull out a few examples here. You guys have access to these slides, by the way. I forgot to mention that. So maybe now's a good time, because I'm not going to go into detail on each of these slides, but we gave you the slide deck. So the best way to find the slides is to go, if you want to open up your browser and go to tutorials.cytoscape.org. Let's see, what have I got here? Can I do that? Tutorials.cytoscape.org, and it should take you to a page that looks like this. The scroll's reversed than mine. And then we're gonna be running through some of these exercises later, so you can leave this tab open if you want. But if you click on presentations, it's here in this opening paragraph. We sort of have a permanent link to the current presentations we have prepared for Cytoscape. If you click on presentations, the second one here, Advanced Cytoscape Automation, is pointing to the slides we're presenting to you today. So I encourage you to follow along on the slides. That way you can look at one a bit longer if we go too fast, or you can kind of skip ahead and read in more detail and go at whatever pace. Again, there's going to be whole sections that we're going to skip because we only have 4 hours. And so this slide deck you can take home with you, share with your colleagues, and go into more detail in the sections we skip through. So, for example, here I want to talk about a couple, I think, key examples of published networks, but only at a quick, what's that? Ah. And if I hit F, I'll go back to full screen. Thanks, Scooter. Okay, so this is an example. So now 8 years old, and it was 8 years isn't that long ago, but it was one of the first examples of this sort of process of exploratory network analysis and visualization going from A, B, C, and D that I think nicely illustrates what we do all the time using Cytoscape, and something you wouldn't necessarily publish anymore now that it's more routine. But they started in A with what we call a hairball. It's this network view that really isn't informative because there's just too much going on there. And through a number of steps, the first step, they annotated the known proteins. This is mass spec data. They annotated the known proteins with these different color categories and just created a subnetwork looking at the connections between only the proteins they knew something about, and threw away all the gray nodes and those edges because they had nothing to say about them. And so that step alone made it such that you can now actually read the labels. It's still pretty cluttered. The edges mean nothing at this point because it's still kind of a hairball. So then what they did is they looked for known complexes. So prior knowledge about which of these proteins form a complex, and they grouped them together in these 3 circles. And that was getting better, getting some more structure that's a bit more informative. And then the last step they did is they collapsed these complexes into single nodes. And now all of a sudden you can actually look at each edge and say something about it and explore this network and interpret it in a way that is impossible here. Same data, nothing changed in the data going from A to D, right? They just integrated prior knowledge and iterated on visualization techniques. And so this sort of process is, you'll do it a lot in Cytoscape. Okay, here's now at a higher level, there's other examples. This is a yeast to hybrid protein-protein interaction experiment, one done for autism spectrum disorder associated proteins. So in red are the baits in this experiment, and then these are the proteins they wanted to know who the interactors were, and in purple are the interactors. And so this was starting to scale up the size of these sorts of experiments in 2011, I think this one was. And so we're looking at 800 interactions and 500 proteins. Here's another example that Scooter Morris was a co-author on with HIV proteins. So this one's, it's done in a human model system looking at human protein interactions with HIV proteins. So viral proteins interacting with human proteins. Again, the baits are the viral proteins in yellow, and the human proteins are integrated around. And then other interactions from other sources were brought in to identify complexes. Scaling up, this is probably one of the larger projects. Here we're looking at genetic interactions. So these are no longer protein-protein interactions, but instead they're through a synthetic lethal screen where you knock out one, knock out another, and then knock out both, and look at the change in growth patterns. You can deduce whether 2 proteins are genetically interacting, functionally interacting, and make a network view of that. This was their first version of it in 2010. This is now scaled up in order of magnitude. So massive collections of interaction data at that level. And so there's a couple other examples I'm going to skip. I'll talk about this last example here. It's a much simpler network. This is just a known pathway, a cancer-related pathway. So pretty simple. It's not that many nodes and a few interactions. But then the complexity here comes in the data overlay. So around each node are different types of data, correlation with mutation data, expression data, and some technique called paradigm. And then each stripe, which you might not be able to see from there, but you could see it in the original publication, is a unique patient. So they have patient data for hundreds of patients, different data types, all on one network view, so that you can see those data in the context of a known pathway mechanism. So that's a nice data integration example. Okay, so there's lots of examples. This page here in the slides is actually a link, and it'll take you, if you click on it, it'll take you to a Tumblr blog that we maintain, where we put a sample of Cytoscape, a few Cytoscape images, created images from the published literature. We add a couple each week. This isn't all of them by any means, but it's quite a few of them. And so just going to this archive and scrolling through it is a nice way to look for inspiration for different ways of visualizing your data. You can search it for the data types that you're working with. Unfortunately, it's also a nice way to find how not to do network visualization, unfortunately. And so hopefully by the end of this, you'll be able to raise the bar. The bar is not that high right now. Reviewers are not doing a good job of rejecting poor network figures. And this archive has a lot in it. So each month, there's about a dozen or so that are added. And this archive goes back, I think, almost 6 years at this point. And so there's a large collection of Cytoscape and network history captured in this blog. So again, if you just say Tumblr and Cytoscape, you'd find it, or the link from the slide deck here. Okay, before we switch topics to Cytoscape and switch speakers, any questions on networks? Hopefully you're all convinced that networks are sort of worthwhile to look at and use in research. Any questions? Great.
Everybody had coffee? Everybody awake?
I haven't had coffee.
Yeah, you haven't.
The coffee break is at 11, and so we'll refill then. Yeah. Sure. How to compare them?
Yeah, if you have two different drivers not done to a network, and one of them sees differential patterns between the two.
Yeah, so just for those in the back, the question was about comparing two networks that might have some of the same nodes and even some of the same edges, but have different parameters associated with them. And we're going to talk about general tools and how to do those sorts of things. We can get to specific problems that you're working on. There'll be time for that in the afternoon hands-on session. And when we come around, we can address specific research problems that you're working on too. But yeah, you should definitely be able to do that by the end of this workshop.
The short answer to that is there's a couple of different apps, and we'll talk about what apps are and how you get to them. They're specific for network alignment. Cytoscape does not do that. Cytoscape, and I'm sort of stealing my own thunder here, but Cytoscape is, the core of Cytoscape is meant to be relatively generic. And then there are a whole bunch of different apps, including ones that allow you to do network alignment. And that's kind of the key word that you're looking for, network alignment.
All right, thanks.
Yeah, you'll often hear us saying there's an app for that.
There's an app. Yeah.
So this is the first case. You got to start it early. Thanks. Was there another question?
Yeah. So can Cytoscape support dynamic networks?
Okay, so you guys are now skipping. You guys are skipping ahead. So we're going to get there. Right now, I just want to get the introduction to networks. We'll get into Cytoscape next, and once we get into the hands-on, we can cover those things. Okay. So now I'm going to hand it over to Scooter and come back for the hands-on part.
Okay, a little bit about Cytoscape. And we're going to get, you'll see a dynamic network sort of is part of this. And there's a number of other things that we can do when we get there. So Cytoscape is open source. What does that mean? It's up on GitHub. You don't like the way something works? You think you can do it better? Download it, change it, send us a pull request. We'll look at it and pull it in. Download it, change it dramatically, and go off and use it yourself. It doesn't matter. It is completely open source. It's because it's written in Java. That is an upside and a downside. It really is truly cross-platform. You can run it on your Linux machine. That's my development environment of choice is Linux. You'll see it on Macintosh today. It runs also on Windows. It's the same, essentially the same file that gets distributed to all of them with a different installer. And it's a consortium. So there's many of the people in the consortium in the room, but fundamentally, all of these institutions contribute to Cytoscape actively. So it's not just one institution, it's several sort of scattered throughout the world. At its core, it's really important to understand the way Cytoscape thinks, heavy quotes around anthropomorphizing an application, but the way Cytoscape thinks about the data model. There are two things that Cytoscape knows about. It knows about networks, and it knows about tables. And to first order approximation, those are completely separate. There are networks and there are tables. Okay? So this is, we think it's a good thing, and you'll see how that all fits together in a little bit. But what you really want to do is you want to take your tabular data, the data that you're going to pull in, and you want to represent that in some way on your network model. And that's done through visual styles. So what will happen is we'll take tabular data and you'll make a style out of that, telling Cytoscape how to treat that tabular data. And then when you map that style in, you wind up with something that looks like this. Okay? So that's really the goal of a lot of what Cytoscape is about. If you think about it, it will have tables, it will have networks, and it will have styles. There's a whole bunch of other stuff that kind of goes around those things, but those are the three key concepts. The fourth concept, which is one that we've already mentioned, are apps. So Cytoscape is extensible. There's a lot of people that have written apps for it. There is an app store. We don't sell anything, but we call it an app store. I tried with all sorts of different names, and I lost.
It's a great kind of store.
Yeah, okay. Yeah, it's a store where everything is free. There you go. At any rate, you can go to the App Store and you can see all sorts of different extensions to Cytoscape. And if Cytoscape is running, if it's not running, you'll see something that will allow you to download an app. If it is running, you can install it while Cytoscape is running, and you've got that app. So in this case, Cytoscape is running in the background. I don't have WikiPathways app installed, or actually I do have it installed, and it's telling me, look, it's already installed. If I didn't have it installed, it would ask me to install it or give me an option to click that button and install it, or click a button to upgrade it. So it's pretty intelligent about all that. There are 300, greater than 300 now, 330 apps. Okay? So that's why there is an app for that. There's probably, as far as network alignment, last count, there were 3 or 4 different apps that just did network alignment. Some are better than others. Your mileage may vary. I think you're going to have to try them out, see how they're ranked. There's a rating scheme for apps. And there's also a link to the citation, typically. So you can go off and you could read the paper and make sure that you think you agree with that algorithm, or it's going to do what you want it to do. So you can also install direct from Cytoscape. There's an app manager. These two things are going to merge in the near future. But right now there's an app manager and you can see all the apps. It's not as pretty. It doesn't give you a lot of the other information, but still sometimes it's really useful. The thing that you can do with the app manager that you can't do with the app store from within Cytoscape is if I have 5 apps installed, well actually, if I have 30 apps installed, which is my general environment, is I have about 30 apps installed. What I'll often do is go through and rather than uninstalling, I'll disable the apps. And that way I don't uninstall them, I just sort of move them off to the side, and then when I want to use them, I'll just enable them. So it's just quicker than downloading them from the App Store. And this gives you an idea on what that looks like. This column right here, in this case it's all installed, but it can also say disabled. Or then you can have the opportunity to enable it. Okay. So how do I get data in? You know, there we got, I've just told you that there's two kinds of things that Cytoscape knows about: networks and tables. Well, tabular data, you know, you can imagine that's pretty easy to imagine how you would get tabular data in. You have your Excel spreadsheet and you're just going to read it in, right? But to a certain extent, we need to map that tabular data onto networks. So the first step is, how do we get a network in? And the easiest way is still with Excel. Typically, when you're going to import a network from Excel, you're going to need a source, some column that you can use as a source node, and some column that you can use as a target node. The target node is optional. So if I only read in source nodes, what am I going to get? Any guesses? What am I going to get if I only read in source nodes and no target nodes? A lot of mumbles. I'm going to get a network, but it's not going to have any edges. I have a whole bunch of nodes. Sometimes that's exactly what you want. And that's a great starting point for a downstream analysis where I'm just going to read in a whole bunch of nodes and have those. I'll create a network through analysis downstream. You can also read the kind of this XGMML file, which is an XML-based network file. You can SBML, there's readers for that. There's readers for biopacks. There's readers for PSI-MI, a number of other different things. And there's a reader for, it's a shout out to Brett back there. There's also a reader for matrices. So if you have a general matrix, you can read that in and create a network out of that. In addition to that, you can go out and you can pull in data from a lot of different public repositories. And we're going to talk about that in more detail later. But Cyquick is built into Cytoscape. The String app will allow you to directly connect to String and pull in all data from String. Reactome, there's a Reactome app, there's a WikiPathways app, Pathway Commons, Index. All of these are apps that you can download. And then once you've downloaded them, you can access that repository and pull in networks from that repository. And of course, the purpose of today's workshop, Cyrest. Tabular data, same thing. You can read it in with Excel. And we're going to go through some examples of that when we get there. And you can also basically push data from R or Python and just, you know, sort of load data that way. Cytoscape has its own session file format. So one of the things that you can do is you're spent hours working on that perfect image. And it's 5 o'clock, and of course we all leave work promptly at 5. Yeah, I saw all the smiles. And you want to save everything, and you don't want to lose all that effort. You just do a save. And when you save in Cytoscape session file, it saves everything. So it'll save the what you currently have selected. It'll save all of your tables, all of your networks. It sort of pushes everything into its save format, and then you can sort of pick back up where you left off. So you can also export images. Obviously, one of the goals here is to get those images out so that you can publish them, put them in your presentations, and Cytoscape gives you the tools to do that. And you can export tables so that you can retain those tables. A lot of apps will add columns to tables. So that you'll start off reading some data in, and then you'll run a bunch of apps, and there'll be a bunch more columns. So there's a good reason to need to want to be able to export that table and save it. All right. So here's what we're going to do.
There's a link at the bottom if you want
do,
If you want to,
yeah.
you don't want to do the browser version.
Oh, I was just going to move
Okay.
you out and do it.
Okay.
So typically what I like to do at this point, you can go through the tour of Cytoscape that's on this link, and it'll give you sort of a push button, oh cool, isn't this cool? And it runs within Cybrowser, and it's great. But rather than doing that, I think it's a little easier for this group is for us to just launch Cytoscape all together and follow through. I'm going to give you, so I'm going to run Cytoscape here. And if everybody follows through, you all have the data that I'm going to use. And that way we kind of learn a little bit about Cytoscape, and then we'll dive back into the slides. Does that sound reasonable? Okay, let's get Cytoscape started.
How do we switch to
We push the button.
push the button? Thinking about it.
That was magic. I got Cytoscape started. I didn't have to do anything. All right, so everybody should see something like this. So everybody got that? We are going to try and follow along. So, okay, so this is the starter panel. What you'll see is these are a bunch of sample sessions, okay, that have been made. But any session that you've previously opened, okay, you're going to see up at the top. So it sort of remembers the last sessions that you've opened. So a couple of different things to note. There are a number of different what we call cytopanels. Often in Cytoscape there will be a CY something or CYTO something. It's kind of the way that I guess it's a branding, I don't know. But there's a number of different cytopanels here. If you look right here, this is where all of your networks will be as you load them. Down here is where all of your tables will be as you load them. The network visualization itself will appear right here. And then obviously you've got a little button bar up here that you can click that will provide buttons. Some of these things may look different on yours because it depends on what apps you have loaded. So the first thing that I wanted to do before we sort of click on any of the sample sessions, because that's just too easy, is I wanted to go through sort of a more realistic example of how you would typically work with Cytoscape. So if everybody can go import network from file. Everybody see that? And it's going to import network from file. We'll bring up a browser, a file browser, and I'm going to go to where Alex installed Cytoscape. Alex keeps a lot of versions of Cytoscape around, apparently. So wherever you installed it, if it's a Mac, it's probably in Applications. If it's a PC or Windows machine, you probably installed it in Program Files. Under that, you'll see Sample Data. So go to Sample Data, and we're going to open galfilter.xls. We're just going to read in an Excel file. All right? There we go. So here's a nice little Excel file filled with, for the most part, meaningless data. All right? Does everybody have that? So what this is telling you as you go through is this right here, the green, is telling you that this is the source node. This is a target node, looks kind of like a cute target. This is the interaction type. And these are edge attributes. So this is going to build a table for us in addition to our network. Now, this symbol, I don't know what this symbol is, but it's not meaningful for our discussion. So I clicked on the column header, and I'm just going to turn that off. I click the little X. And I'm going to do the same thing for value. So everybody should see exactly that right now. Good? A lot of shaking heads. Then, lo and behold, we're going to say OK. And we've now created a network. That's it. Now, our network isn't terribly exciting. So now that we're in the network, though, a couple of things to note. First of all, this little guy down in the lower right-hand corner is providing you with an overview of the entire network. So I can grab the outline and drag it around and see where things are. But in the network itself, I can zoom in and zoom out, either using the plus minus, or if you have a scroll wheel on a mouse, you can use a scroll wheel on your mouse. If I want to select a node, I just select it. Just click on it. As with many other things, if I do Shift Select, I'll add to the selection. And hopefully you noticed as you did that, down in the table panel at the bottom, it changed to just show you the rows that correspond to those nodes. Now, in our case, that's not really informative right now, but we're going to make it more informative in just a little bit. If I want to select a bunch of nodes, I can click out. That deselects everything. I can just hold down Shift and drag and sweep out a bunch of nodes. All right? So a lot of different ways to get in, to begin selecting things. Often let's see, here's a good example. If I have a hub node, you can see that that's a hub because there's a lot of different edges going into it. And in this case, if people can find YNL216W. If you can't find it easily, another little trick. If I type YNL216W in the search bar, it'll find that and select it. Okay? Once you have that selected, often what you'll want to do is I want to expand that selection. I want to find all of its neighbors, and I can do that very quickly by saying select nodes, first neighbors, undirected, or Apple 6 or Control 6 on a PC. So very quickly, I can start building a subnetwork. And you can see how now I'm starting to do, I've gotten something loaded, and now I'm doing some analysis of that. Still rather unhelpful. I don't happen to know what YNL216W, not being a yeast biologist, it's sort of opaque to me. But we have some more data that we can bring in. Now what we're going to do is we're going to say File, Import, Table. Remember I said there's networks and there's tables, and now we're going to add to this table from File. So File, Import, Table, File. And this time we're going to bring in galfilter.csv. And this looks a lot like what we saw before, right? Same kind of interface. Oh, wait a minute, wrong file. That's just a, no, sorry. File, import, table, file. You can hit cancel on that one. And it's gal expdata.csv. There we go. That looks a lot better. So what this is telling us is this is a key column. It's going to try and map that to the data that's already in the table. In this case, it's exactly the right thing. If you can see, we have the same kind of identifiers here as we have here on shared name. And this is telling me what column it's going to try and match it to. Okay? And everything else is right. It's just going to import it. So I'm just going to say OK. Great. Now I have a bunch of data. Does everybody see that? Everybody have their data in there? Great. So remember, my goal here is to help out the poor non-yeast biologist because I do not know what YNL216W is. So I clicked on my Style panel. And now we're going to take information from our table and we're going to map it to our network. And the first thing that I want to do is I'm going to come down here and I'm going to change label. And you can see that the column that it's using for the label is name. I'm just going to click that and change it to common. And lo and behold, my mystery protein or gene is RAP1. All right? And now from here on, you can see that the columns, that common name is being used for all of the data. Everybody got that so far? Okay, so we'll do one more thing. I'm going to do two quick changes, and then we'll call it done there. First thing I want to do is I really don't like the rectangles, so I'm going to change to ellipse. I'm just changing, I'm at the leftmost column right now, so I'm just changing the defaults. Okay, so now I've got ellipses. The very bottom of the style, you'll see lock node width and height. Now I have circles. Okay? And then the last thing I'm going to do is a little more complicated. At fill color, I'm going to click and I'm going to open up. I'm going to click the little triangle. I'm going to open that up. And I'm going to change the fill color. I'm going to use a column, gal1rgexp. That's, this is the gene expression data. It's fold change data. And then I'm going to change the mapping type to continuous mapping. And now, very, very quickly, I can see the fold change. Okay? So that was just a really quick tour through some of the things that you can do with Cytoscape. You've seen, I didn't use any apps in this case. I could certainly have brought in a number of apps. We could do analysis of this network. We can actually calculate the degrees of each of the nodes or the degree centralities of each of the nodes, size the nodes by those degree centralities. We can bring in other kinds of information. I can access this data from string. A number of different ways to spread this out. And we'll have the opportunity to do that. And you'll see some of that when we're driving this from RAS, from R, in just a little bit. All right. Quick tour. By the way, typically we spend half a day on working with tutorials on Cytoscape, but we want to get to the R stuff. Any questions before we jump ahead? Overwhelmed? Not sure what to do with this. Yeah?
And like kind of a technical question. For the sessions, so they almost serves as version control itself.
You could think of it as version control. I could save, but each time I save a session, I'm going to overwrite it.
So it's better to just get single sessions.
Right. So typically what I do is I just overwrite the sessions. I usually don't save multiple sessions unless what I'll often do is if I've made a lot of changes and I know I kind of like it, but I want to try 5 or 6 other things, I may save the session then so that I can roll back. And because of the starter panel, if I can just kind of bring it back up and say I want to go back to this version and then just click on it. Okay? Yeah?
Under the property, there are 3 columns. One is default value, one is mapping, one is bypass.
Correct.
Yeah, what do they mean?
What did they mean? We're going to go through that, but let me just quickly tell you that this is a mapping, okay, where we're mapping data from here onto the network. The default value, you saw that we changed, so for example, we changed the shape to a circle. That was a default value. Bypass is where I have a network, a particular value, and you saw that in the example that Alex worked through, where some had squares, some were round. That could be done with a mapping, but maybe what I really want to do is, we'll go back to our friend RAP1. RAP1 is important. It is the purpose of my figure. I want you, audience, to see RAP1. That's what I want to do. So what I'm going to do is I'm going to come down here and I'm going to click the bypass value. I have to select RAP1 first, and then I'm going to change my bypass value to 100, and RAP1 is going to stand out. Right? Similarly, and this is another example, another trick, is what I would also do generally if I really wanted RAP1 to stand out, is I would change, again, I've selected it first, I would change the bypass value of the border to 4, I would change the color of the border, and you can see all of a sudden, even though the expression is relatively low, I can really highlight this in my network. Now, you would want to, in your figure legend, RAP1 is made larger so that it stands out, however you want to describe that. But you can see very quickly I can start styling this network and making it visible. Everything that I've just done, you can do from R. Okay? So everything.
Another way to, I often think of those 3 columns, default, mapping, and bypass, right? They override one another as you go from left to right.
That's right.
Default,
Good point.
default's the default, that one makes sense. Mappings will override whatever you said is default if there's a mapping, and bypass will override either defaults or mappings if you set them appropriately. So it's a way of just sort of customizing from left to right each property.
Sorry, quick question.
Yeah.
Is there a quick way to revert everything back to default?
So you can, there's a little trash can icon, you can delete individual mappings to revert everything back to default.
Yeah. So let me show you one thing about styles. Just like in Excel or in Word, where you can have a set of defined styles, it's the same in Cytoscape. These are styles that are already predefined. So I can, if I really kind of want to just play with a style, I'll take the default style and I'll copy it. And then I'll just play with it until I get what I want, and then I can just delete it. I can just delete the style, or I can drop back to default. And often what I'll do is I have 2 or 3 networks I'm playing with. I'll set up a style for each one, or sometimes I'll set up a style for the same network. So one of the things that you'll hear me talk about real soon is not to overload your networks with visual styles. And so sometimes I'll have 2 or 3 styles that will focus in on the part of the network that I want for this figure. Okay? All right. Back to the slides. Let's see. This is the tour of Cytoscape. If you open a tool called CyBrowser, which is the built-in browser, you can go through this tour. I don't think we need to do that. We're just going to jump ahead because I think everything that's in the tour you sort of got. Okay, so the next thing I want everybody to kind of understand is not all networks are the same. And you want to think about these networks differently, both in terms of the analysis that you're going to do and in terms of visualization. So before we dive into analysis and visualization of networks, it's really good for everybody to have a sort of a common taxonomy of networks. And the first one that I want everybody to think about is pathways. So everybody knows what a pathway is, conceptually. We can have metabolic pathways, the Krebs cycle, or TCA cycle as it's now called. We all learned that. Some of us had to memorize it. Signaling pathways, regulatory pathways, all of these you've seen in networks. Now, look at this pathway. This does not look like a protein-protein interaction network, does it? It's characteristically different. And it's different in a couple of different ways. One, it's abstracted. This is not all of the interactions that these individual proteins go through. We've thrown away intentionally a lot of data here that we know in order to show this particular cycle. There's also annotations of various kinds. These three, we have a box around them. We have a number of just names coming in that are annotations. So this is, pathways tend, when we bring them in, they tend to be curated. They tend to be abstractions. They're focusing in on some aspect of what's going on in the cell, not everything. Another example, this is a signaling pathway. You can see the same kinds of things. You can see that I've got the little phosphorylation. Very it almost looks like a circuit diagram. Everything's laid out very carefully. And my best example is this. This is a pathway. Now, when you think about this pathway, everybody recognizes this, right? I hope. It's the London tube map. So when you think about this particular pathway, there's some things that should be obvious. One, not all transit in London is on this pathway diagram, right? If I put all of the buses and all of the other things, the trains, all of the train stations in London, it'd be a mess and it'd be completely useless. Similarly, the other thing about this is this is not what the Thames does, right? This is an abstraction. I've abstracted things out to make it easier to understand. And so when you think about pathway diagrams, they are diagrams. And I think very carefully I term them as diagrams. All right. The one that you're probably most familiar with are interactions, protein-protein interactions. This is the canonical and ubiquitous hairball. That's the official name for this type of visualization. We do not publish hairballs anymore unless there's some very particular unique aspect to it. Protein ligand diagrams, though, right? There's a particular, this is showing basically all of the ligands that are interacting with a particular protein. There's a whole method, the C method, where you can start looking at the similarity between proteins based upon the similarity between their ligands. And that's being used heavily right now for drug repurposing. So it's a big, pretty hot area. Domain-domain interactions is another type of interaction diagram that you can see. A protein itself is a network, right? If I take a look at this protein, I can take a look at each of the amino acids and plot as a network the relationships between the amino acids. Either the hydrogen bonds, the covalent bonds, salt bridges, I can get all of that in there and treat that as a network. And all sorts of others, up to and including social networks, which is probably the canonical example that we're all aware of. Oh, and yes, the wine and cheese network, very important. And this just came up, and this is one of my favorites. This is a Cytoscape network that showed up in the Tumblr blog, and it's apropos of what's going on in Russia right now. And that is, this is a network analysis of a youth soccer team. And you can see that what they've done is they have the closeness score, the closeness centrality, and we'll explain centralities in just a little bit, is mapped to the size. These are two different teams. And you can see the betweenness score and the passing density on the edges. So a very clear, nice, concise representation of how these two teams differ. What I didn't read the paper well enough is to know how that affected their scores. That'd be interesting. But at any rate, the other kind of network is similarity networks. And similarity networks, the sort of canonical example here is a protein similarity network or a sequence similarity network. And these are incredibly useful for annotation transfer, functional annotation transfer. Okay, if I look at this cluster right here, everything that's this tan color has known function. Everything that is gray has unknown function, right? So I would be pretty confident assigning this function to this protein based upon this analysis. And in fact, there's a website called the Structure Function Linkage Database that's doing exactly this kind of thing. But there are others, chemical similarity, where we're looking at Tanimoto or Jacquard coefficients, ligand similarity, which I mentioned earlier, and topic maps. Kind of the non-biological example is where you can take two authors and see how similar their works are. All right. Yeah. So any questions about any of that before we start talking about sources? And we're going to kind of skip through sources really lightly. Any questions about the taxonomy of networks? Kind of makes sense? A little bit? No? I want to see what this means when I get to Cytoscape. I'm holding my judgment. That's what I kind of, okay. All right, sources. So how are we going to get data in? Where does this data come from and how do we interpret the data? I would suggest going through these slides, and I'm not going to. But basically what I'm doing in these slides is going through a whole list of experimental techniques. I want to hit the top of the techniques. So I'm going to kind of fly through them and just mention them. But in particular, all of the content behind the slides, the speaker's notes, include a lot of detail about these techniques. Fundamentally, the idea here is yeast-2 hybrid. How many of you are familiar with yeast-2 hybrid? Great. You know how it works. Here's a nice pretty picture going through how it works. But the basic idea is I'm going to use a bait, and I'm going to use that bait to find all of its interaction partners. Great. Another similar technique is APMS. It's a very similar approach. Again, I'm going to use a bait. I'm going to tag that bait. I'm going to push that bait in. I'm going to pull out a number of different interaction partners and then analyze those using mass spectrometry. BioID is very similar to APMS, except you're not looking for binding partners. In BioID, you're looking for proximity. If it's within, depending on which technology you use, if it's within roughly 10 nanometers, you're going to tag it, and then you can pull it down and you can say that this was close. Basically, again, pretty pictures to tell you how it all works, and a whole bunch of different. BioID is the new hot area, or proximity-based labeling is the new hot area. And this very wordy slide says that there's been a whole bunch of different modifications of that technology. And then there's just some of the protein fragment complementation assays, a slower approach, not as high throughput, phage display, protein microarrays, chemical cross-linking, and this is really the slide I wanted to get to. So this is old, 2008. It hasn't been updated, and I'm going to talk to the author of the slide this week and see if we can do an update to this slide. What this slide tells you is when you're looking at these sources, these experimental techniques, they're not all good for the same kinds of binding partners. So these techniques are different, and they'll pick up different things when you're looking at these bait-prey approaches. So if you're looking at TAP-MS and you have a series of cytosolic proteins, then you're in good shape. But if you're looking at very hydrophobic proteins, you're not going to be able to pick those up. So you have to understand what kinds of negative values, potentially false negatives, you're getting when you're looking at these various experimental techniques. There's a number of, another genetic interaction techniques, completely separate. So a great way to confirm binding is to look at the genetic interactions and the protein-protein interactions and make sure that you're getting a similar signal. And that'll give you a pretty good evidence that you've actually got data here. I'm not going to go through this. And then there's a whole bunch of low throughput techniques, okay, which you all should be familiar with. And then computational techniques, text mining. How many, if you think about it, for these low throughput, where does that data, careful biochemical experiments that show binding, what database does that data show up in? PubMed. Very rarely do these careful analyses where you're doing specific binding assays wind up in any of the major databases. They just get reported in a paper. So some of these text mining techniques may be the only way that we can extract some of those low throughput technique data. Orthology and, of course, domain pairs. All right? So, great. Now you know something very quickly. Now you're all experts in the realm of all of this binding. How do we get that data in? Where do we go find the network? Right? I want the canonical network for my data. Where do we find it? There's no canonical network. I just told you about a variety of different experimental techniques, right? So there isn't a single network. In fact, there isn't even a single repository. There are so many different repositories for different databases that provide different things. It really depends on what your question is. And we're going to guide you through a couple of different approaches in some of the analysis, but we'll give you pointers to a number of different network repositories. So here's 3 examples. I'm going to talk about string in particular, mostly because I wrote the string app, so it's my preference. But the idea here is that these are very broad coverage, and they're not really specific. They're lower resolution. So let's take a look at the string app and what you can do. So string has genomic context predictions, high throughput lab experiments, conserved co-expression. It has automatic text mining. That's one of its main features, is the text mining that you get out of string. And it also pulls data from other databases. And if you have the string app installed, you can get string protein query. So you're just going to take your list of proteins, you're going to click string protein query, you're going to paste them in there, and you're going to go and you're going to get a network.
Should we do this as hands-on?
Yeah, you want to show that?
Yeah. And so they could start by installing the string app?
Yeah, does everybody have string app installed? It was part of the, so let's start with there.
Okay. Okay, so we're going to start at the App Store to make sure you guys have the string app installed. The one way to check that, and also the source for it if you don't have it, is to go to the App Store. So that's apps, apps.cytoscape.org. And then if you just search for string, you can type string in the search bar in the upper right. And it might be a little slow today since we're all doing it at the same time, but it should show up near the top there. The string app, this is what it looks like.
Hey, 5 stars.
And yeah, there's a rating. Got 25,000 downloads and 5 stars so far. So let's click on that and you'll see a dedicated page for the App Store. And then it should tell you.
If you have Cytoscape running.
If you have Cytoscape running, which, yeah, if you all kept it open. Cytoscape App Store and Cytoscape, your running instance of Cytoscape are communicating with each other, and it'll tell you if you have it installed or not. If not, this will say install, and you'll click on that, and it'll install it in your running session of Cytoscape, and you should be ready to go. So that's it. That's the one click. Yeah.
Technical question.
Yeah.
What do you mean what data it's communicating?
Yeah. So the website there's a dedicated port in the Cytoscape in your Cytoscape application to communicate with the website. This website. Yeah. If you want more technical details, we should talk after.
Yeah.
And so yeah, it knows about what you have installed and what you don't have installed in the current state. If it's out of date, it'll tell you that you can update it. So it's a nice way to just use the website to manage your apps. So now back to Cytoscape. What you should see, here we're left off where we were from the earlier hands-on demo, fine. And in this upper left section of the network panel, maybe you're in the style panel and you need to go back to the network panel, you'll have this search bar. And in the search bar are some built-in, index is built in, SiteQuick is built in, and then there's app-driven options. All of these four that have this string and stitch icons, those come from the string app. I happen to have the Wiki Pathways app and Gene Mania app installed, so you might not see those, that's fine. But go ahead under string, I'll let you choose. You can either pick disease query, in which case you'll type in your favorite disease. Favorite's a weird word to use there. Or use the protein query and type in your favorite gene names or the genes that you're studying. And so, Scooter, do you have yours, or does someone want to yell out their favorite genes?
I have my cancer list: EGFR, BRCA1, BRCA2, TP53.
That should all sound familiar. Great.
Yeah.
53. So you can type them in. New line is the default. If you're copy pasting these from somewhere that has semicolons or commas or something else, you can specify that. I clicked on this 3 bars here as a configuration settings for this search bar, and here you can choose. What I was saying was, if you're going to use delimiters other than new space, you'll want to check here, use smart delimiters. I'm using new space, so I don't have to. And then here's where you'll specify the species that you're querying, and it auto-completes for you because there's a lot of them. So if you just start typing musculus, you'll get musculus. If you don't, you'll have to scroll through here forever to find musculus. So there's a lot of species supported by string, so that auto-complete's important. Let's go ahead and just stick with
Homo sapiens.
sapiens. And then there's 2 really important things for the string app. There's the confidence cutoff. The default's 0.4, which is pretty lenient, meaning that 1 means there's great confidence for an interaction, and 0 means if it's in the database at all, give it to me, I guess. And so 0.4 is the default, which is pretty good. If what you get back is too much of a hairball, there's too many interactions, you can always dial this up to like 0.7 or 0.8. Those are recommended levels. And then this additional interactors is a nice feature. The default 0, meaning it's just going to find interactions between our 4 genes of interest. But say if we bump this up to 20, we're saying go ahead and add 20 additional interactors that might, maybe there's not a connection in between BRCA2 and TP53, but there's an interaction between an intermediary. This will allow bringing in that intermediary and additional interactions that expand beyond just your query set. And so we'll go ahead and choose 20 for this demo, and then click the search icon, and now it's querying the string database live and pulling back all of the interactions between my original 4, and I'm going to use that nifty search tool that Scooter was showing to highlight them. There's my 4, right? I just pasted it into the search. Here are my 4, they're highlighted now in yellow, and all the interactions around them. You can see this is an example where maybe there's too many interactions. All I got was this tightly connected hairball. And so the easiest way, I could redo the search, or under the apps menu, there's some string options here. and under string, you'll see change confidence. And I can just change the confidence score. This is the same slider, and just let's make that really stringent. I want like around point, I can tile it in, 0.8, 0.8. Say okay, and it's gonna just remove all the ones that were below 0.8 in my network. And I can redo the layout. Still pretty tight cluster. So that's a string.
Right. And that gives you an idea, that whole interaction, those steps, gives you sort of an idea on what can happen with a lot of the other apps. They all add this kind of both menus and buttons and things like that. If you're interested in pathway data, you can use Wiki Pathways, which is a great app for bringing in pathway data. Again, Alex showed you that. It's another app that you can download. And the thing here in Wiki Pathways is you can import it as a pathway or a network. And the strength of Wiki Pathways is the pathway diagram. And you bring in that pathway diagram with all of the annotation that comes with it. Okay? Any questions about that? Yes?
And so, what do you do when you have like a text file of proteins? You want to make it like how do you import that instead of just copying and pasting?
So if you want to go to string, okay, then you need to copy and paste. Okay, so but you can copy, so you can read in, you can do a read in, and you can now, remember I said that you could, sometimes what you wind up doing is you read in a list of proteins and you build that as, without any data associated with it, with no edges. Often what I'll do is I'll just read in a list of proteins as a starting point that have a lot of information about those proteins. Then in the table panel, I'll copy and then I'll paste it into string. And I'll build the network that way. All right.
We have time. Should we do index?
We have a half hour, right?
Yeah. Okay.
We haven't done the network. I want to make
Right.
sure that the,
For Viz.
yeah.
Okay.
Yeah, I think. Okay. Any other questions before we, yes?
I'm assuming all of this can be done through R and
Everything that we've showed you so far can be done through R.
And we're going to do it right after the coffee break.
And you were going to have to load the string app anyway for what you're going to do afterwards. Yes. Apps can export their capabilities out to R and Python.
Python 2?
Hmm?
Python 2?
Yeah, I said that. I did say that.
Oh, one more question.
Yes.
Yes. From the network that we extract from the setup sheet that you just show us, can you have us to extract some information from the network from string?
So if you, are you still up?
Yep.
Like from the color, what can we
There's
learn
a-
about the color of the egg?
That's a good question.
It is.
The color is meaningless.
Not the color of the edges.
Oh.
The density of the edges.
Of the edges? So sorry. I was talking about the color of the nodes. The color
Right.
of the nodes is just a pretty style choice
It's
that
random.
string.
It comes from string, just like you would get on the string
That's how the
website.
string website does it.
Just like the little.
There's even
specular
structures in
highlight.
there
Actually, brief anecdote here.
that are real structures.
Yeah, the structures actually come down from string. I've collaborated or known the string folks for a long time. We teach a class together every year. And I kept saying, we really need a string app in Cytoscape. And for the longest time, it says, well, we really can't do that. And finally, we sat down and said, why? And it's because, well, Cytoscape can't do glass spheres. And that's part of our branding.
They didn't want this.
And I said, oh, I can do that. Oh, okay, then let's make a string app. There we go. So the glass spheres are important. That's string branding. And if you look,
But no, there's a lot of good stuff.
take a look at this. I'm getting all of the compartmental information. I'm getting tissues. I can get Pharos data. Basically, and if you looked at the edge table, here's the score that we use for our cutoff. But here's the individual score. Databases, experiments, co-expression, text mining, co-occurrence. I have individual scores for those edges that I can go through. And if I want to sort of throw away any where I don't have experimental evidence, I can highlight everything that has an experimental evidence below a certain value and throw those edges away. All right, so I have a lot of data. And I guess we'll go ahead and show, there we go, show the side panel.
Which one, sorry?
Apps, the string side panel. So apps, string, show results panel. And get rid of diffusion. That's a bug that's now fixed. So in addition, you get link outs that you can click on, and that will go directly to the tissues database or to gene cards or whatever. There's a lot of data there. And I guess we'll do the final thing. We might as well do the whole thing. Apps, string.
This one?
Yeah.
So separate.
Separate. Click out.
Oh, sorry.
We don't want that selected.
Oh. The node?
Now, yeah. And string has functional enrichment, so we provide functional enrichment. And you can download your functional enrichment. Okay? And color the network and do all sorts of things. Yeah, just go to there we go. So there are better charts than that, but Alex likes pie charts. I tried to convince him that they...
I just clicked the link. I just clicked the button.
You have the default set.
Oh.
Yeah. Okay, so that gives you an idea on how much, and it's not just string, right? Pretty much all of these apps are giving you a very rich environment for pulling in data from these resources. Index, anything that was in the index, the posited network from index, you're going to be able to pull back. You saw wiki pathways, all of the data that comes down with the pathways. All of these repositories are out there, and they try to provide you with a great deal of information. Okay, so now you have all that data in there. Come back. What can you do with it? I'm going to really go through the analysis approaches pretty quickly. I want to get to visualization. Hang tight. There's some key analyses that I think are critical, and I'm just going to hit those. The key one is, if you look at it, degree is, we use degree as a proxy for a hub. So degree is just the number of edges, the number of neighbors of a node is the standard. You can have in degree or out degree if they go in different directions. The betweenness centrality, these are centrality measures. So you can have degree centrality, which gives you an idea on how hubby this is. The betweenness centrality gives you an idea on how many paths through the network go through this node, right? So you can, and then the closeness centrality says how close this node is to the rest of the network. So now, if you think about it, what happens to this network if this node is perturbed and no longer binds? The network splits in half, right? So you would sort of think, if this was a full network, that would be, if we're talking biology, you would think that that's an essential gene. Similarly, you would think that this is an essential gene, because a lot of things bind to it. Now, there may be other paths, so it might not be in the case of degree centrality. Closeness centrality basically says that a perturbation of this gene is likely to flow broadly throughout your network because it's close to everything in the network. Doesn't mean that it's essential, but it means that it's close enough that that perturbation might flow out. So if you think about those, you can think about all of these various topology statistics. I can think about modularity, motif analysis, and network alignment. All right, so a couple of quick concepts, and then we're going to move on. So a lot of what we do when we're doing network analysis is graph theory. And mathematicians don't call things networks. They call them graphs. They're the same thing, but they like to use, I don't know why, N is an overused symbol, and they use G. And if you read papers, and I guess that was just a better symbol. Similarly, they're not nodes, they're vertices or vertexes. All right, fine. And then you can think about an edge or a link. We both call them edges. The only thing we agree on is it's an edge. If you think about a network, this is a great sample network for illustrating a number of different things. A key thing to understand, because it's what Cytoscape uses, is a multigraph. Cytoscape allows multiple edges between nodes. Now, in biology, this is really important because the interaction between two nodes might be different, or I might have multiple types of interactions between these two nodes, right? So they might both be part of a complex, but separately they might do some sort of modification. So I think in Cytoscape we allow you to have multiple edges, and those edges can have different types. A hypergraph says that an edge can connect more than two nodes. Cytoscape does not support a hypergraph. That's not in its network model. This is a quick and dirty hack. There is a node of exactly 1 pixel right there, which is how I did this example. Okay? But it is not a true hypergraph model. I measure a couple of quick things about network measures. We can look at shortest path. Look at the slides if you want to get that. The key thing that I want to get to is how we typically will look at degree. This is obviously a string network, and we can show the degree by changing the node size to map to degree. Again, the degree becomes a column in the table, and now I can set up a style that will say, okay, I want the degree to set the node size. And in a network like this, it's really easy to see, the larger, the higher degrees nodes when you size them. But it's a dense enough network that if I didn't size them, sitting here and counting edges, 1, 2, 3, 4, 5, 6, 7, 8. Oops, I missed one because this doubles up, so there's 9. You can see that it's hard to do that, which is why we typically will use these sizes. A number of different network measures. I don't see those reported that often anymore in the literature, in the biological literature, but just know what they are. And here's clustering coefficient, but the one that I want to get to is, here's the 3 centrality measures, and this is how they're calculated. Now, the easiest way to calculate these is to count the degree of each node and then, the easiest way to do it is to use an app to do it. And there is actually a built-in app called the Network Analyzer, Tools, Network Analyzer, and you can see that it adds columns to the table, and then once you have those columns in the table, we can use them and reflect them back into the network on size or color or whatever we want to do. Okay? I think that that's it. I think I'm just going to
Those are links now.
jump to here.
If you want to skip, those are links now. If you want to skip to a section.
Oh, cool. The slides changed between a week ago when we gave this last time, and I'm getting told, hey, look, beware, the slides changed. Okay. Very quickly, the last section here before our break is on to, yes?
Sorry, before you move on.
Mhm.
So there are apps for all the analysis you mentioned, or they were just examples?
All of the analyses that I talked about, you can get from the Network Analyzer. You don't even have to go to the App Store.
Oh, okay.
Okay, the Network Analyzer will give you all of those measures. There is also another really really good app called Cytoscape, or Cytoscape, depending on if you're Italian or not. And the author is Italian, that's why we've been corrected. It has a large number of centrality measures, which is a great app, and it's called Cytoscape. You can find that in the App Store and load it. Yeah.
Are there times when it's easier to do the analysis outside of Cytoscape, like in R with another tool like iGraph, versus having to load the whole network and have like the visualization active while doing it?
So I should mention. Okay, cool. That's what happened behind me. Yes, there are, and it depends on the analysis that you're doing. However, what I will say is none of these analyses depend on that network visualization, the network being visualized. So basically, in Cytoscape, if you bring up that string network, kill the, and he's got the
Switch it back to your favorite.
Yeah.
What do you want to see?
Go ahead and kill the view.
Oh, I see.
Oh, sure.
Destroy the view. So I still have the network, I can analyze that network, and often when you're going to read in huge networks, it won't even try to create a view. You can do the analysis, you can use the select tab to do sub-selections of that, again without a view, and then I can create a new network and that'll give me my subnetwork. And that's typically the workflow. Now sometimes if you're taking advantage of an R package that does a very particular kind of analysis, let's say that what you have is you have single cell RNA-seq and you want to run it through Seurat before you do your downstream network analysis. Absolutely. Do that in R, get your, do your aggregation of your cell types and then feed that in the Cytoscape for the visualization. Okay. Other questions? Yeah, sorry.
I have two questions. The first one is, how can I say the information such as the degree or closeness to apply and use later?
Right. So as I said before, you can save tables. So if you calculate the degree using the network analyzer, it puts all that data into the node table, and I can just export that node table and read it into Excel. And then, or as you're going to this in an hour, a little less, you can just have an R connect to it and pull that data, pull the degree column into R, and then use that for an R analysis if you want to. All right.
And
So, yeah.
And the second one is how large is the network that the NAT can handle or compute the degree?
How large is? Yeah. So Cytoscape it uses a lot of memory, although we're getting better. We're working really hard on that. So the how large depends on how big a machine you have. So for example, I have done MCL clustering using the Cluster Maker app on networks that have tens of thousands of nodes and millions of edges. Okay? On a machine that had 128 gigabytes of RAM. And no, that wasn't my desktop, unfortunately. I think you have to know sort of what you're doing, but it does take a lot of RAM. And some algorithms, which are recursive in particular, can get just really bogged down with very, very large numbers. You have to be careful. Occasionally what you'll do is you'll run an algorithm. If it really starts slowing down, and in Cytoscape, at the lower right-hand corner of Cytoscape, right where it says HDMI, no, right there, you see this little memory? That's green, and this will give you the free, and it gives you basically a tracker for memory. If this starts going yellow, then you need to get more memory or simplify your network. Okay? All right. So there's a number of ways that you can show a network. But, and that's in the slides, and I would sort of, you know, if you want to get some ideas on how to visualize networks that aren't the traditional node link diagram, as it's called, I'm giving you a bunch there. But since right now we're really going to focus on Cytoscape, I'm going to skip that section since I have links now, and go to data mapping. So Cytoscape and most actual network visualizations allow you to map data values to a number of visual attributes. Now the visual attributes include node fill color. You saw me change that when I did the quick walkthrough. Border color, border width, the size of the node. In fact, the width and the height of the node, the shape, the opacity, the label. Often I can put charts and graphs, as you saw with the string app. I can change the edge color. I can change the type, the width, the ending type, what arrows I have, what starting type, the ending color, the starting color. All of these various parameters that I can get to, I can change. I can do it in a couple of different ways. I can use a continuous mapping. So when you saw me put up that gradient, that was a continuous mapper. Any numeric value, okay, I can use a continuous mapper to map to some of these visual attributes. Typically, I would use continuous mappers for size. I would use it for color. Okay? I can use categorical or discrete mapping. They're called discrete mappers. And then what I'll do is I have some sort of categorical data. I can map that to shape. I can map that to color. Shape is a stronger signal than color, keep in mind. So depending on what you want to do, you want to think through what kinds of mappings you're using. And then pass through, which is typically labels, and you saw me do that, where I just said, I want this column to pass through and be the label. If I had a column, so for example, I can use a pass through for size. If I have the size of a node in a column, or something that I want to use as the size of a node in a column, I can use a pass-through and just say, fine, use that. Okay? I can use, Cytoscape also supports X and Y values. So in R, if you're doing some sort of very sophisticated node positioning, then you can export that as columns and map those as pass-through mapping in Cytoscape, and it'll just move the nodes to where you want it. So all of these various mappings and all of these three mapping types. So let's go through it. So I just mapped the name. I'm now changing the edges. Whoop, gotta fix this.
It's doing that for me too. I
Yeah.
don't know why. Yeah, it just wants to skip that slide. We have some timing thing messed
Yeah.
up there.
Okay, at any rate, that's where you get to, and you can see that you get this really, you can, each individual step, and I'll let you run through it. There's a bug in the slide set that we're using that just wants to go right through there. Okay, so this is my, yes, I really am this old. I used to work for a pharmaceutical company called Demchak. It was right there in the Silicon Valley area. And one of the head scientists at Demchak was really, really into Apple. And when the Macintosh was first released, okay, we got a bunch of them. We had a little postage stamp screen, it's not that big. And it was really cute. It kind of sat on your desk, a little floppy disk you put it in. But there were two things about the Apple Macintosh that made it really fun. It had a program called MacWrite. And MacWrite had this feature where you could change the font family. Okay? This was new, just to let everybody know this wasn't common at the time. It was a long time ago. And you had these laser printers that would print these font families out. So we got a whole bunch of Macintoshes in, and for about 3 months, every document and 6 font families that they were just absolutely unreadable. They were it just, it's like, I don't know what I'm supposed to look at here. And then eventually, people settled down and started realizing that it was great to use font families for highlighting very particular things. And they would switch the font families for headers, but not in the middle of the text. Don't do that with your networks. If you can change the size, the color, the shape, you can change all of that stuff. And often you'll want to do that when you're at the exploratory stage. Don't let that network out of your computer. Nobody will understand it, and what you'll do is you'll confuse your readers or your viewers. Okay? I've seen networks on the Tumblr blog where you look at the network and then you look at the figure legend. And the figure legend is so long because they've changed so many things. Or you'll look at the network and so some nodes will be red and some nodes will be green. But nothing in the figure legend that tells you why these nodes are red and these nodes are green, right? But you should never have red and green, by the way. Everybody knows that, right? You don't have to map the phosphors, which is how we started with red and green. Color blindness, there's about 8% of the male audience that use red and green. So if you must use red and green and shape, vary both. Okay? And then you can get away with it. But other than that, you should never just use red and green and expect your viewers to understand that. So anyway, please, if you need to, you're trying to communicate 2 different values. Okay? Sometimes the easiest way to do that is in a figure, you have 2 networks. Change the style, then you have 2 networks. And the good example in that figure of the soccer team, right? There was another image right underneath that. And in fact, even in that image, I could probably have generated the same image that he had with the 2 teams in a single network. I have enough power in terms of the visual attributes to change both of those at the same time. But it would not nearly be as clear as the network, the 2 different networks that he was showing, and it wouldn't be as compelling. So keep that in mind if you need to create multiple network images. We're actually working on a paper right now called the 10 Simple Rules for Network Visualization for PLOS Comp Bio, and hopefully that'll come out pretty soon, and that's one of the main ones. It's actually the one I'm writing right now, so it's... At any rate, layouts. A couple of different layouts. The grid layout. This is often this will kind of be what happens when I read in all of those nodes with no edges. I'll just see it as a grid. Not really useful for much, other than just to, particularly if I'm just varying some single color value, I can just kind of get an overview. Yeah, there's not a lot of data here. Hierarchical layouts. This is a hierarchical layout, but it's not hierarchical data. Don't do this. Right? If you have a tree, if you have hierarchical data, or you have a section of your network that is hierarchical, that's a great time to use it. You don't have to use the same layout all the way through your network. You can highlight a bunch of nodes and change it. Circular. So here's an example where this is degree sorted. We've changed the size of the nodes based on the degree and the color. So you can get a real nice overview in a small network. There was another Tumblr. The Tumblr blog, by the way, is a great place to say, to look at a particular network visualization and say, I do not know what this is trying to tell me. That tells you not to do it that way. And there was a network visualization that had probably 2,000 nodes in a radial layout like this. And then a little cutout of a highlight that blew up, like 20 or 30 of them. And it's like, what is this telling me? And at any rate, what you normally think of as a force-directed layout. And a force-directed layout is pretty simple. All you're doing is you're saying these are masses and these are springs. And then you're doing a physics simulation. And all of the force-directed layouts essentially work the same way. They all give you this mass and string. Now, keep something in mind, right? This is an n-dimensional space, and you're compressing it down using an optimization into 2 dimensions. So if this is a weighted network, it is not the case necessarily that the length of this edge will be precise in comparison to the length of this edge if these are different weights, right? Because you're having to do this optimization to get it all down to 2 dimensions. And then, as I mentioned, you can combine layouts, and there are lots and lots and lots of layout algorithms. Now, the thing to keep in mind with layouts, and sometimes you want to tweak. I want to take 5 nodes, and I want to just move them over, or I want to align them. And of course, you have that capability also. And that's under the node layout tools, node layout tools, and it brings this little dialog up in the network panel. So what's the purpose of layouts? Use layouts to there's 2 real reasons. One, I want to emphasize the relationship between the nodes, or I just want to stretch things out for aesthetics. You need to understand what it is you're trying to say with your layout, and then use the layout that's sort of appropriate. Layouts can be tuned. So in Cytoscape, layout settings, that will bring up a dialog box, and then you can choose the layout that you want. And that'll give you all of the little knobs and dials that you can tweak for the length of your spring, the strength of your spring, the mass of your nodes, all of those kinds of things. And you can change that and click the Apply button, and then change it again and change it again. I'll also say one of the really nice things that Cytoscape has is layouts are undoable. So if you do a layout algorithm and it's like, oh my gosh, I just went 2 steps backward, you can undo that layout and get back to where you were. And then the last in depiction is animation. So getting a little bit at time series data or dynamic networks. Here's an animation that was created using Cytoscape. And in fact, at the end of the day, there we go. I was about to say, this is playing on my screen, but it's not. Okay. At the end of the day, there's an app for that. It's called CyAnimator. And what you're looking at here are 2 networks, actually 1 network, that had 2 different types of edges. Remember I said networks can have 2 edges. And what I did is I deleted all of the edges of a particular type and did an MCL cluster, and then added, and then I switched and did an MCL cluster the other type. And now I had 2 network visualizations, and I used CyAnimator to interpolate between the 2 network visualizations. Now let me tell you what the data is. So, wait for the next one. The one that it's going to right now, that one, is measuring the similarity. This is a similarity network. It's measuring the similarity using BLAST. Right? The other one is measuring the similarity using C. So what we're saying there by the other one is that 2 nodes, 2 proteins are similar if their ligands are similar. Right? So the dogma, the classical dogma, is sequence implies structure, which gives us function. Right? And the similarity in ligands is an analogy to function. So I've got sequence similarity in one network and functional similarity in another network, and they should cluster the same. Right? Any guesses what's going on here? So this was a collaborator, Brian Scheuch kid asked me to put this together, and I did it. And he looked at this, kind of stared at it, and he looked at it, and he said, oh, okay, yeah, that makes sense. And he walked away. And I said, wait a minute. So, think about it for a second. Blast is doing an entire sequence, right? It's typically the binding domain that's giving you this functional specificity. So, if I look at this, and I haven't done that analysis and I'd love to, but what I think you're going to see is a lot of the ones that are very, very different are multi-domain proteins where most of the domain is the same, but the binding domain is different, right? And now I have a different specificity. So when I look at the blast similarity, these things are very similar, but in fact, the specificity will be very different. So, animation can tell a compelling story. One of the things that you should be looking at whenever you're going through and thinking about your visualization is what's your story? What is it that you're trying to communicate? What's the science behind it that you want to get across to your audience? And this is one that has this really compelling story behind it. It's very easy to tell that. You need to have that same kind of thing for your network visualization when all is said and done, and you're going to put that in paper. When you're exploring your network, map whatever you want onto it, right? Because it's still at the exploratory stage. You don't know what the story that you want to tell. But as soon as you get to that story, you want to have fewer nodes, fewer edges, fewer changes in terms of color and size. All of the ones that Alex went through were relatively simple networks. In some of those cases, I saw the original data, and they weren't simple at all. They had to go through a step to analyze those and pick out the subnetworks of interest. All right.
Question?
Yes.
All of this cool work and all of the work that you went through to generate that animation, is it possible to generate an animation like that from R and Python?
Yes. Cyanimator is an automated app. So fundamentally what you can do is there are commands in Cyanimator where I can build my network in Cytoscape and I can say, remember that frame. And I can build another network in Cytoscape and I can say, remember that frame. And then I can say, animate between those frames. So, yep. Okay, other questions? Alex, yeah, go ahead.
This Cytoscape can be used as a sort of a split mode with one version running in an R server and Cytoscape and the desktop actually doing visualization.
No, it can't. Now, one of the things, so the question was, can Cytoscape be used in a split mode, separating the front end from the back end, essentially? There's a number of apps which actually run on the back end. String. You know, the string functional analysis is an example which we already went through. Diffusion. There's a diffusion app that's a core app in Cytoscape that actually runs on the back end too. More and more apps are going to actually run their algorithm on the back end and just use Cytoscape for visualization. But fundamentally, not all of them do, and not a large subset. And it's not architectural in Cytoscape. There is a framework for doing it, but
And does it have an RStudio-like mode that is entirely done within the browser on the server?
no, it does not. So the question was, does Cytoscape have an RStudio-like mode where Cytoscape is run entirely in a browser? And the answer is no. It's all written in Java. And that's sort of one of the limitations to Java Swing. Other questions? Right?
And the coffee's here.
Exactly 11 o'clock. I want you to note on time.
I'll definitely recommend coffee because once you get back, it's going to be all automation. So we're going to be
Yeah, we're done with most slideware. Now we're going to walk through automation walkthrough, and then you're going to have lots of time to work on your own projects. Or